Hello everyone, my name is Lillian Capados, and today my group members Ira Bates, Madeline Oliver, and I will be discussing our project titled Natural Microcosms as Useful Models in Ecological and Evolutionary Research, Tank Bromeliads, Pitcher Plants, and Rock Pools. Our research begins with understanding what is a microcosm. Microcosms are small and contained ecosystems that can be utilized to test ecological theories in a natural setting. Shrivastava et al. 2004 explains to us why these systems are utilized to test ecological theory. Natural microcosms are small in size, tractable to do manipulative experiments, highly replicable, have natural environmental variants, and realistic species combinations. Our research focuses on the natural microcosms tank bromeliads, pitcher plants, and rock pools. Tank bromeliads, researched by Ira Bates, are plants in the pineapple family that trap water in their centers or tanks. These depend on the aquatic food web, including insects and microbes, to decompose litter and provide nutrients for the plants. Bromeliads do this by absorption through their leaves. Pitcher plants, researched by myself, are carnivorous plants with pitfall traps that are used as a mechanism for trapping prey and secreting digestive fluids. Common American pitcher plants, Sarracenia, are seen in many parts of the United States. Pitcher plants trap thousands of insects such as ants, flies, and even wasps. They're home to small aquatic communities of microbes and arthropods. Lastly, rock pools researched by Madeline Oliver are isolated pockets of seawater found in intertidal zones of oceans. This is where the ocean meets the land, in rocky ledges or sloping sandy beaches. These small basins capture many organisms such as snails, barnacles, mussels, urchins, sea stars, small fish, and many more. This information brings us to our research question. Are natural microcosms increasing in importance as models to test general ecological and evolutionary theory?
We reviewed the literature to document how natural microcosms are used in ecological and evolutionary research and how their importance as model systems has changed over time. To perform the review, we then conducted a literature search by combining keywords in the web of science such as tank bromeliads and models and community. This was performed to identify relevant scientific papers published between 1974 and 2020 that utilize these natural microcosms to test ecological theory. Every paper resulting in each search of bromeliads, pitcher plants, and rock pools were analyzed by reading their abstracts and determining if they were relevant to our research questions. Papers dealing with this natural history of our systems or descriptive papers were excluded from the results. All papers deemed relevant were then sorted into categories based off their ecological scale of study, either organismal, population, community, or ecosystem, as well as their main theory their research was based on. Web of Science search results yielded 343 results for tank bromeliads, 281 results for pitcher plants, and 336 results for rock pools. We averaged the total results of all three microcosms to show their importance along the entire ecological scale. We found that the entire literature search, 8% of papers were organismal level studies, researching physiology, thermoregulation, and evolution. 14% of papers were population level studies, observing species abundance, metapopulations, and population growth. Community level studies averaged 51%. Observing species interactions, meta-communities, and island biogeography. Ecosystem level studies averaged 27% of papers found, studying nutrient cycling, organic matter, and decomposition. Overall, community level studies were the most common study done based on the ecological scale. Once categorized, the totals of included papers for each individual microcosm were calculated to percentages based on the ecological scale ranking of included papers. For all three microcosms, community-level studies seem to be the most significant studies in paper testing ecological and evolutionary theories with natural microcosms. Order of importance for each ecological level was community being the largest category, then ecosystem, population, and lastly, organismal. This is seen only for bromeliads and rock pools. Pitcher plants yielded different results. Figure 2 shows the data for pitcher plants and suggests that organismal level studies in regards to the microcosm were just as relevant as ecosystem studies. This is something that we did not see with rock pools or bromeliads. Organismal level studies in pitcher plants outnumbered population level studies, which we did not see in bromeliads or rock pools either. The order for pitcher plants is different, going from community, tying ecosystem and organismal, and last, population. Figure 4 shows that there is a steady increase in relevancy of microcosm utilization in papers testing ecological and evolutionary theory. Our results are based on a 46-year time span, from 1974 to now. We identified that from 1974 to 1997, bromeliads were uncommon as model systems, but have now become the most important natural microcosms used in ecological studies of our selected microcosms. Rock pools and pitcher plants are about equal in importance and parallel each other in a steady increase in use over a span of time. Our results make it very clear that tank bromeliads, pitcher plants, and rock pools are valuable natural microcosms that benefit research in ecological and evolutionary theory. We conclude that these ecological communities tend to be studied more using natural microcosms because there are simple structures and ability to test complex theories. Natural microcosms are useful systems to utilize when testing ecological and evolutionary theories, and their importance has shows an increase over time.
We would like to acknowledge the Gonzalez Lab. Thank you for listening.